excited to share with you our research from Dr. Hensley's cell biology lab from this past semester. Over the last 2 years, the biology department has been reinventing the way that we learn. Instead of going into a lab with a recipe-style lab manual, following the directions and getting an expected outcome, the students are going in and getting genuine research experience because they are able to design, create, and execute their own research experiments. One way to get the students involved and invested in these types of labs is by choosing topics that are interesting to them and relative to their everyday lives. Our lab decided to work with CBD in various cancer cell lines. CBD, which is a non-psychoactive component of the marijuana plant, has become increasingly popular over the past couple of years. Forbes did a study comparing relative search volumes of CBD and other topics in the mainstream media. CBD is in green on the graphs, and as you can see, it's more popular than the World Series, Taylor Swift, and even Jesus himself. With the popularity of CBD increasing, the topic of marijuana legalization has also come to the forefront. Here we have a map of the different levels of legalization in the United States. The dark green states indicate complete legalization, and the lighter states show varying levels of legalization. In Alabama, we are the lightest shade of green, showing we have no legalization whatsoever. This is important because as college students, we're going to be the ones making the decisions about this plant's future in our state. So with all of that being said, my group decided to work with Charlotte's Web CBD oil and melanoma cancer cells. We decided to use melanoma because it's the most dangerous type of skin cancer, with survival rates as high as 98% when it's localized, all the way down to 23% when it has spread through the body. In order to increase our survival rates and patients' quality of life, further study and therapy options need to be researched. One area of interest is the use of cannabis and its derivatives. We decided to use Charlotte's Web CBD oil because it's medically reputable. It was created by the Stanley brothers in Colorado for a young girl who was suffering Dravet syndrome, which is a type of childhood epilepsy. Our hopes is that the CBD oil will decrease cell viability and migration. The first test that we performed on our cells was a viability test called an MTT assay. For this, we had 5 separate experimental groups: an untreated control, a sunflower control, and low, medium, and high CBD dosage groups. We plated our cells on a 24-well plate, and 24 hours after being plated, we administered the drug treatment. And then 72 hours after being plated, we added the MTT chemical. If the cells were alive and respiring after the drug treatment, then an enzyme in their mitochondria will cleave the MTT chemical and create a purple precipitate. The intensity of the purple can then be quantified with a spectrophotometer, and its absorbency can be recorded. Our hypothesis is that CBD will decrease cell viability. Here we have the results from our MTT assay. On the graph's x-axis, we have our drug treatment groups, and on the y-axis is the cell viability. The first 2 columns that you see are our control groups, and they're dark purple because there were viable cells that were stained. And as you can see, when we added the increasing dosage of CBD, the viability of the cells decreased, showing that CBD decreases cell viability. The next test that we did was a migration test called a Boyden chamber assay, and I actually brought one in here. So for this, we have these special chambers that we place the cancer cells into the top part of this, and there is a semi-permeable membrane at the bottom of it. So we put the cancer cells in the top with a nutrient-deprived media, and then we suspend them over the wells with a nutrient-rich media in the bottom. So in theory, these cancer cells would be able to rearrange their cytoskeletons in order to move through the membrane so they can reach the nutrients. We want to see if the CBD will decrease the migration. And here we have the results. On the x-axis are our drug treatment groups, and the y-axis is the percentage of migration migration. The dark purplish blue that you see is actually a high concentration of stained cells that were able to make it through the membrane and down into the bottom well. And again, here you can see as we increase the concentration of CBD, the migration decreases, showing that CBD decreases cell migration. In conclusion, we found that Charlotte's Web CBD oil decreases cancer cell viability and migration, and our data suggests that the use of CBD in cancer research warrants further study. Also, because of our experience, my group will be presenting our research at the Association of Southeastern Biologists annual conference, and Dr. Hensley's graduate seminar hopes to expand on this research to submit a publication. Thank you.
So if you said this, I missed it, but since Alabama is so light green on this, what kind of hoops do you have to jump through to get the CBD for the research and is it just available, as long as it's for research, it's just available and you use it? Or?
Well, so CBD is not illegal, so you can get that in all 50 states, but if we were to try to use other chemicals like THC or AGA,
So yours is just the derivative for the CBD?
Mm-hmm. Okay. We would have to get like a DEA license.
So your 40 micromolar concentration, your high concentrations that you use, how high is that concentration? Is that something that's relatively low or high, or would normal cells be affected by that concentration? Or do we know?
I'm not completely sure. I know that we had a solution that was 60 or 70 mg per mil, and then we diluted it into the concentrations. But the tincture that we used was from a website that's safe for people to just ingest it. So.
Dr. Raven. So at my previous university, when we did research with purified CBD, we also tested it on myoblasts and osteoblasts as they would differentiate into osteocytes, and we saw significantly less cell death. There was some, maybe 10% versus 85 to 90% in the cancerous cells. So it doesn't seem to just generally affect dividing cells, because we specifically chose undifferentiated cells that were rapidly dividing. And so it does not seem to affect normal cells the same way that it affects cancer cells at these concentrations.
Any more questions? All right, let's thank our speakers.